Model, MEM, 2011. Portable mammogram breast cancer diagnostic device. Atlas, for multi-frequency electrical impedance mammography. Distributed by Innovation Medical Devices. Dedicated to discovery, committed to care.
Tomography, Greek T. Section, cut, is a method of non-destructive layer-by-layer examination of the internal structure of an object through its numerous transillumination in different cross-sectional directions. Methods of tomographic examination: X-ray computed tomography, magnetic resonance computed tomography, radioisotopic tomography, ultrasound tomography, positron electronic tomography. Methods of impedance examination: rheography, body composition analysis, bioimpedance spectrometry, electrical impedance tomography. Method of electrical impedance mammography. Method of electrical impedance mammography. The 256 electrode electrical impedance mammograph, EIM, was developed by the Institute of Radio Engineering and Electronics of the Russian Academy of Science for realization of the method of electrical impedance mammography. Russian technical condition 29442-002-711888036-2008. Conformance certificate. ROS. RU.IM 04VO6814. Registration certificate. FSR 2008/02744. MEM electrical impedance mammograph is a specialized system for electrical impedance computer tomography. It is intended for the diagnosis of pathological changes in mammary glands, tissues and other near-surface regions of the human body, by measuring the relative change in electrical conductivity of biological tissues in cross-sections of the organ under examination at different frequencies, and by visualization of distribution of electrical conductivity on a PC screen. Electrode matrix use for measurements. In order to implement the method of electrical impedance mammography, we have developed a measuring system, and an algorithm for reconstructing images that use a set of electrodes arranged in the form of a two-dimensional matrix on a flat surface, to visualize static distributions of electrical conductivity of the media adjacent to the surface with electrodes. Method of electrical impedance mammography. During the examination, the matrix is retained against to the mammary gland towards the ribs, so that maximal quantity of electrodes is in contact with the patient's body, and the thickness of examined tissues is minimal. To pick up electrodes, the first one is used as general electrode of current source, and the second serves as reference electrode of potential difference indicator, are joined between each other and are situated on the patient's wrist.
Remote electrode. Electrodes of matrix. Equipotential surfaces. Lines of electric current. When the current flows through a medium, it creates volume distribution of the electric potential. The potential decreases along to the current line in proportion to moving away from the electrode. Injecting the current. Loss of voltage per unit of length is proportional to the current magnitude I, and to the resistance of a medium R, according to Ohm's law. Measuring the loss of a voltage V, and knowing the current, it is possible to calculate the resistance value. The voltage measuring instrument is connected to the first electrode of a matrix by a multiplexer, and then the current source is connected serially to each of the remained electrodes of the matrix and measurement is carried out. After that, the cycle is repeated for another registering electrode. The complete set of the data, used for reconstructing of three-dimensional distributions of electric conductivity, consists of 65,280 measurement results. The data handling and initiating of the measurement process is carried out by the personal computer, connected to the instrument by Universal Serial Bus, USB. Power supply is also carried out through a standard USB port. For reconstructing of three-dimensional distribution of electroconductivity of a medium, there is used the method of weighted back projection alongside with equipotential surfaces of electric field. Reconstructing of electrical conductivity is carried out in several tomographic planes, cuts, parallel to the electrode matrix. It is necessary to mean that though this system provides static visualization, i.e. visualization of spatial distribution of absolute electric conductivity of the tissue, it does not allow to determine absolute values themselves. There is reconstruction of electrical conductivity deviations from the mean level for the given patient, and not its absolute values. Therefore from now on values of electrical conductivity defined by the system are expressed in relative, or standard, units. Mammary gland, structure, ultrasonic scanning. Nipple. Peripapillary muscle. Lacteal sinus zone. Anterior folium of the split pectoral fascia. Subcutaneous adipose tissue. Cooper's ligaments. Durate crest. Glandular tissue. Lacteal ducts. Posterior folium of the split pectoral fascia. Retromammary adipose bursa. Electrical impedance tomograms at different planes of scanning. 50 kHz. Electroconductivity of biological tissues. Biological tissue consists of cells and intercellular space, filled with substance electrolyte. Internal content of a cell is separated from intercellular space by a membrane, which equivalent electrical scheme represents itself parallel connection of the resistance and the capacity. Thus, electroconductivity of biological tissues depends on the frequency of passing current and the form of its oscillations. Some biological tissues are able to answer on passing current by excitation. In this case their electroconductivity nonlinearly depends on the current amplitude. If there will be no excitation, then currents are disturbed into tissues in accordance with the impedance of its components. Cellular membranes represent relatively big resistance for the low-frequency currents. That's why their basic part passes through intercellular gaps. The amplitude of the low-frequency currents is proportional to the volume of intercellular space, for example, to the lumen of blood vessels, and the concentration of electrolytes in it. Measurement of electroconductivity of biological tissues on such low frequencies are used in biology and medicine for definition of blood filling of different organs, revealing of edema of organs, in which swelled cells decrease intercellular space. Electroconductivity of biological tissues, measured on high frequencies, is proportional to the general quantity of electrolytes, maintaining in the tissue between the electrodes, so in this case cellular membranes haven't already hindered to distribution of electrical current.